because they're walking around and you can notice them. But what we don't see the effect on are the smaller things or the things we can't see beneath the surface. And that's why when Avery back in the 17 realm.
Hundreds?
Yeah. 2017, we thought one thing that might be useful is if we get some baseline data going for the harbor. We contracted the Center for Coastal Studies to come out and do bathymetric mapping to look at the state of our bottom in the harbor, do some sediment grabs, and it showed us a lot. It showed us we have some real healthy areas here in the harbor, mainly due to our tidal flow. We also have some areas that are pretty bad, mainly because of excess nitrogen, because people coliform, E. coli, all kinds of things that are showing up increasingly because there's more of us here on the Cape. So we thought, while we've got this snapshot relatively fresh, let's get a snapshot of who's swimming around out there. Both the finfish and the shellfish. And it'll give us some data that we can look from going forward. Maybe what the findings will show that we need to resample in a few years or want to for a particular species of concern. But it's everything from the mummichugs and pupfish, all the way up to the stripers. We're all waiting for it. And no one better to speak to that than our lead researcher for the finfish study from the Center for Coastal Studies. Owen Nichols is with us tonight. His invertebrate Agnes Mittelmeyer is the lead invertebrate researcher. She couldn't be here tonight, but Owen's going to fill us in on how this timeline of the study is going to unfold for us, maybe what we can expect, some of his previous work. And our thought is that once he finishes his run of slides, we can take a break, a quick intermission to grab some cookies and coffee, and then we can kind of open it up for discussion on what's interesting to you. And all of you have seen so many of these faces here out on the harbor. Owen might have questions for you. Where should I sample? Where would be good? And I'll turn it over to him. I will say that getting world-class help and research done, like getting an organization like the Center for Coastal Studies to actually do this study, it's again world-class, but it's not cheap. And since I'm going to let at the end of this presentation and discussion, I'll let everyone just filter out. I will say now we are certainly looking for help to fund the study. You're all grassroots. If you're not familiar, we're all volunteer with the Friends of Barnstable Harbor and have been since we got together to come up with a fix for the bulkhead that collapsed many years ago now. But for now, I'm going to turn it over to Owen and let's hear about what's going to go on.
Well, thanks for having me. Can folks in the back hear me okay? I'm used to screaming over a diesel or an outboard, so hopefully I don't need to turn this on because I really don't like microphones. But yeah, thank you so much for the introduction. I really want to thank the Friends for supporting research in Barnstable Harbor and particularly supporting this study. This work builds on a foundation of benthic habitat mapping conducted by Agnes Mittermeier and her team, as John mentioned, and I'll circle back to those maps at the very end. It'll be a kind of a good starting point for discussion, I think. Our general plan, we hope to get underway as soon as the Friends give us the green light as far as funding goes and so forth. But basically, we'll be doing monthly surveys. We'll try to do a full year of surveys of Barnstable Harbor, and I'm going to go through some work that we've done in Pleasant Bay and Nauset Harbor, kind of give you a sense of the kind of work that we've done in the past and the kinds of information we can collect. Very loosely, we're going to apply some of the same methods that you'll see here to Barnstable Harbor. That said, if our discussions today lead us to start thinking about some novel methods that maybe we haven't tried in other places, I think there's a lot of value to that too. We do try to standardize some of what we do so we can compare across different systems. We can compare Nauset to Pleasant Bay to Wellfleet, Barnstable, things like that. But in general, I think I'll just kind of jump right into it. hopefully you'll kind of get a sense of the flavor of the work that we do. I'll tell you just a tiny bit about what I do at the center. So believe it or not, this will be my 25th year at the Center for Coastal Studies. I grew up out here on the Cape. Currently, I direct their marine fisheries research program, and much of the work that we do is in collaboration with commercial fishermen, shellfish farmers, recreational fishermen. And basically what we do is try to answer questions that the community has. So I hope you have some questions today. You know, we don't go into our research with preconceived notions. You know, we really try to keep an open mind and ask questions that are of interest to everyone in the community. And again, my work focuses on working with the fishing community. The center as a whole, we like to say that we do science that matters. It's, you know, it's a pretty catchy phrase, but we really do want to do things that matter to folks like you, to all the stakeholders around Barnstable Harbor. And so please, you know, hold on to questions if you have them at the end. I also don't mind if you need me to clarify something, just raise your hand. But I hope we'll have a lively discussion at the end. For folks who have to cut out early, if you need to run, I left a few business cards at the front desk there with our website on them and my email, so please do reach out. All right, I'm just going to jump right into it here, and we'll see what happens when I press the button. Slides don't always work. So I'll talk a little bit about our work in Pleasant Bay. This is kind of where this kind of work really started for us at the center. And just like Barnstable Harbor in Pleasant Bay and later in Nauset that I'll talk about as well, we started our work with the benthic habitat map. I'll say one of the parallels here is that, you know, there's a long history of human exploration, of harvesting natural resources. In the case of Pleasant Bay, Champlain noted that there were a lot of bay scallops and oysters, and I do like to look back through the historical literature and the older studies that were done. That's a big part of what I do. You know, John used the word snapshots at the beginning, and that's something I'll circle back to as well. But, you know, looking back through time at some previous snapshots is really important. And folks, if you know of any sort of historical literature, older papers, especially the ones that are kind of hard to find, that talk about the natural history of Barnstable Harbor, things like that, I love hearing about those. But I'm going to talk a little bit about our approach to studies in Pleasant Bay. You know, certainly Pleasant Bay is well known for an abundance of winter flounder, quahogs, and these are just shots from Brian Gibbons' logbooks, one of the commercial fishermen that spent a lot of time in Pleasant Bay. You know, again, we build a lot on the work that and the local knowledge that the fishing community has. So these are some logbooks that were shared with us from the 60s and 70s. In the case of Pleasant Bay, there was also this sort of foundational study that was done by the state of Massachusetts back in 1965 and 1966. They did a series of these studies, these marine resources studies, and there was one done for Pleasant Bay. They did a pretty comprehensive look at fish communities, shellfish, and one of the recommendations of their report that was published back in 1966 or 67 was that we should do it again in 10 years. Unfortunately, that hadn't been done, and the state really hasn't replicated any of these studies. They did one in Salem Sound in the late 90s, early 2000s. They did that with private funding. Unfortunately, the state does not have the money to do this work. And so we've been doing it with funding in most cases from private partners. In the case of Pleasant Bay, and you know, like many of our harbors and bays around Cape Cod, they have what's sort of a little bit unique in that, you know, the bay is, you know, the watershed of 4 towns around it. They have a natural resource management plan. It's been updated, I think, at least once since I made this slide, not twice. But the Pleasant Bay Alliance has this management plan for Pleasant Bay that largely focuses on water quality, but also makes a lot of recommendations on other things. And one of the things they've been pointing out for the last few iterations of the management plan is, you know, we know there's been a lot of changes, a lot of variation in abundance and when and where things are in the bay, and with effects on commercial and recreational fisheries. And, you know, we really ought to be doing some research on the habitat, on the commercial and recreationally important species in particular, but also to do that to develop a framework for long-term monitoring. And that's going to be something we'll come back to as well at the end. Snapshots are great, and we hope this is really valuable. But we really see these inventories as a tool with which to develop a framework to look forward, just like John said. Another Friends group, your counterparts, the Friends of Pleasant Bay, did something very similar where they had a fundraising campaign to support a really comprehensive assessment of this system. They funded some benthic habitat maps in partnership with the National Park Service. They funded the project that I'm going to talk about here. And because we have a lot of seals in Pleasant Bay, they supported some research on that. And we'll talk a little bit about the fisheries work that we did. This started with a kind of a pilot project back in 2014. And this is kind of cool, speaking of changes in resource abundance and quotes and things like that. Bottom right, a gentleman named Charlie Beggs, who had a long history of not only owning a mooring company in Pleasant Bay, but also harvesting shellfish. And he sent me an email back in, I don't know, 2013 or something, and it was titled, "You Don't Know What You Got Till It's Gone," you know, the Joni Mitchell song, right? So I opened this email, and he's talking about lobsters in Pleasant Bay. And one of the things that he noted, again, the value of observations and community knowledge, you know, working in the mooring business, every time he would haul his moorings in the fall, he would notice that there were lots of little juvenile lobsters around these moorings. Now, Pleasant Bay and Nauset are both areas where in the past, and cyclically, there's been a high abundance of lobsters, enough to put at a minimum a few kids through school and things like that. But we also had wondered if Pleasant Bay and Nauset are kind of a nursery for lobsters. And so Charlie really kind of pushed me to start working in the bay. So we started with this pilot study just focusing on lobsters. We set out these passive collectors, and we put these in Cape Cod Bay. We'll be putting these along the eastern shore of the Cape Cod in the coming years. But we put them in Pleasant Bay back in 2014, and just a couple of spots around Big Bay here, down towards Quonset Pond and down towards where Pleasant Bay Community Boating is now. They're basically boxes full of rocks. You drop them in early summer, you haul them up again in October. They're like miniature artificial reefs. They've got a fine mesh tray that you can't see inside this wire mesh that's like lobster trap material. Fill it up with cobble with different sized rocks to create lots of little interstitial space for critters to hide in. And then you haul them back up at the end of the season. We then, and I'll circle back to the results of that, but we then expanded the study starting in 2015 using a variety of other research methods. This is something that we'll almost certainly be doing in Barnstable Harbor. This is our 60-foot seine net. This is a commercial grade beach seine. It's I got it from Memphis Twine down south. This is what bait fishermen use further south, but this is also the same type of net that the state used when they did their studies in the 60s. And so we wanted to use the same gears to have a comparable result. And we focused on a couple of sites that the state had surveyed back in the 1960s. And you'll see this is kind of a recurring theme where we try to go back to sites that have been studied previously with our similar gear whenever possible. So we would go out every month and hit these stations a couple times each month and try to be as standardized as we can relative to tide and weather and things like that. We also did subtidal surveys. So we were building on one of Agnes's benthic habitat maps. So all these little dots are places where Agnes had done her benthic surveys. We picked a subset of these to go back to. And we also picked a few sites that DMF had surveyed back in the 1960s. So again, the sort of hybrid approach of building on some of the benthic work, because it is really good to have the linkage between the samples that were counted or collected during the benthic mapping relative to our work. So it's nice to, at least in some areas, go back over where Agnes went. But we also used previous studies to kind of guide where we went. In this case, we were using the Shackleton. That was the pride of the fleet at the time. And we went out and, again, monthly surveys using a couple different types of gear. One of the gears we use, again, in a very standardized, replicable way, is a lightweight base scallop dredge. This is also an awesome benthic sampling tool. And again, working with commercial fishermen, in this case, Ted Lucas, who is now our marine operations manager, but when he started on this project with us as a commercial fisherman from Chatham as well, this is one of the tools that he had in his garage, and we absolutely found it really, really useful for benthic surveys. We're looking at macro benthic animals. So Agnes does the micro with her grabs. I do the macro with a dredge. We also used a 30-foot sweep otter trawl. It's basically a Wilcox shrimp net. It's a nice piece of gear. The same gear that DMF used in the 60s and more recently in Salem Sound to repeat their work there. In this case, it was literally the same net. Since then, I've had Rydars down in New Bedford building a new one to the same specifications. This net's taken a beating, but we still have it. But again, monthly surveys using standardized gear, trying to go to the same spots each month. So building a seasonal picture of how things change over time. In many cases, we also use ventless lobster traps. Again, another standardized gear. These are basically like relatively small commercial lobster traps, minus the escape fence to let the undersized lobsters and crabs go. We want to catch everything with these traps. So some of the results from the Pleasant Bay work really quickly. So when we hauled those collectors back in the fall of 2014, we did see some juvenile lobsters, and in fact, we had the highest density number of animals per unit area in the state at the time, which was really cool. To be fair, you're creating a sort of an artificial reef effect by putting this box of rocks and mud in Pleasant Bay, so it's probably a little bit elevated relative to what you might see in the absence of those cobbles. But it was really nice to see a high density of juvenile lobsters there. But we also saw a lot of fish. And we had a lot of the usual suspects. Anytime you have structure like that, you're going to see millions of little cunner and other species. But what was also really interesting is we started seeing tropical fish. Here's some examples of what we found in these passive collectors. That's a snowy grouper, and that's a butterfly fish. And even growing up on the Cape, I'd seen these butterfly fish here and there. If you threw a minnow trap in the right space in the summertime, you might see one of those. But we also saw these little snowy grouper, which was at the time a new one for me. I hadn't seen those before. When we got into the seine sampling, and I'll say, this was kind of the first thing that we did, and this was really exciting because this kind of leveraged a lot of interest in doing the expanded work that we ended up doing. People got excited that we saw such fish diversity, even just putting these passive collectors down. Seine sampling, again, sort of the usual suspects, the mummy chugs, the killifish, lots of small pogies. This was kind of when we had the beginning of that big run of small peanut bunker, the little tiny menhaden everywhere that started kind of in 2014, 2015. We were seeing them in Meeting House Pond and Pleasant Bay on Thanksgiving. They were really abundant, and we've seen that consistently almost every year since, which is really nice. Lots of Atlantic silver sides and other bait fish. We'd see a lot of fish in a single net haul. When we pull that seine in, this is all seine data. One set, we had almost 3,000 of these silver sides at a time. So you have to work quickly. This is a great place for volunteer support. Many hands make light work. We also saw fish like this needlefish here. That's a really cool species. We had the aquarium down collecting a few to take to Boston. A great forage fish for striped bass and other species. Basically, we saw a lot of bait, as you might expect using that type of gear, but it's really important. That's the base of the food chain as far as a lot of species go. And so this is a great way to get a good sense of what bait species there were out there. As far as the trawl and dredge surveys, what I'm going to do, I'm not going to throw many numbers at you here, and I'll argue sort of throughout this talk that the numbers are interesting, but not all of it by any means. The number of species, I would argue, is more important than the number of animals for just a one-year snapshot. But to use one of our subtitle sites where we trawled and dredged and trapped, it's a eelgrass and sand area. There's a eelgrass bed, there's sand, there's some macroalgae here. This is in Little Bay, a little bit to the north. A variety of fish species. That's a tomcod, lumpfish, some bay scallop seed. So we're seeing some species of commercial and recreational interest or importance. Lots of crabs, of course, lots of spider crabs, some big male spider crabs there. That's in Pleasant Bay, of course, we've got lots of blue crabs, and they've been historically abundant there. This is one that was actually shedding, molting. So you can see it coming out of its old shell right on the light board there. And of course, lots of green crabs, in this case, a female with eggs. We'll probably talk a lot about green crabs today. They've become an unfortunate hobby of mine, so we'll talk a little bit more about green crabs. We also now, when you move to the southern here, down to Crow's Pond. So this is down, if any of you are familiar with Pleasant Bay, this is right near Eastward Ho, one of the big golf courses. Again, a little bit different species community. Lots of these round objects at the bottom of Crow's Pond here. Took us a while to figure out what they were until you look really closely. You can see Callaway written on them there. We also had squid in there, long-fin squid. And they were laying eggs inside the Crow's Pond right here, which was awesome to see. One of the downsides, though, Crow's Pond is noted for having really low oxygen. It's an area that probably suffers from a fair amount of nutrient pollution. The whole bottom of this pond is kind of covered in this anoxic black mayonnaise. It's very anoxic sediment. lots of tunicates over the top, which make this carpet of biology over top of it. It's really sour bottom. And sadly, these are freshly laid squid eggs that are starting to die and blacken because they're sitting on this anoxic muck. So we do learn a lot about the habitat when we're out there. I will say this is another species that was interesting to see. These are seaboard gobies. These are really cryptic fish. You often don't see them when you trawl, but if you dredge and you haul up some empty shells, or in this case, these things love old beer cans, find a lot of seaboard gobies in empty beer cans and things like that. And these gobies really don't care. They're like the Oscar the Grouch of the fish world. You know, they only get about this big. They'll find them in old beer cans, and they don't mind the low oxygen. They actually do really well in that. So getting a sense of what can survive in places like this, especially given those anthropogenic stressors and things that can't, like these unfortunately squid that probably won't make it to hatching. When you get down towards the inlet, of course, again, a whole other species community. And again, these are just little snapshots. Lots of winter flounder, small winter flounder. Some really dynamic mussel beds that we saw down here. Sand eels, sand lance out towards the break. So that trawl net actually, despite the slow speed, is remarkably good at capturing some of these forage species, some of the faster moving forage as well. Lots of crabs, again, using those ventless lobster traps out near the inlet. We would set these traps for 2 or 3 days. I cheat a little bit. I use river herring that I get from the state that in turn, when they sample river herring for science, they have these racks, these frames. Is this being recorded? It is. Oh no, that's okay. It's fine. So river herring makes really, really good bait, as you'd imagine. So they typically give me the leftover fish when they're done with them. So nothing goes to waste, which is really nice. But they make, as you can see, they'll make a trap like this plug right up to the top. In this case with rock crabs, but in other places with green crabs. These are a little more meat for weight. But that's at the time my intern Tim realizing that he's going to now have to measure all of these crabs, and he's not super happy. So just to quickly summarize the work in Pleasant Bay, you know, we counted thousands of organisms. You know, we were able to survey throughout much of the year. We saw some general patterns of abundance. When I say abundance, I mean relative abundance. You know, month to month, greater and lesser. You know, we're not doing fishery stock assessments here. We're not calculating how many fish there are in all of Pleasant Bay or Barnstable Harbor. We're looking at what we call relative abundance. You know, are there more or less in this area or that area, or this time or that time? And that's really, really valuable. I would argue it's in many ways more relevant than just how many there are. But looking at seasonal patterns of abundance, relatively similar to what the state saw in the 60s, although there's a notable difference in species abundance, species composition. We use slightly different fishing gears. But again, when we tried to compare the comparable ones, there were some notable differences. First of all, we only saw what are essentially young of the year winter flounder. We didn't find any big fish, even though we used exactly the same trawl gear and same methods. And that's a pattern that's not unique to Pleasant Bay. That's going on elsewhere as well. We think that's in part due to warming water temperatures and that the fish are not spawning inside anymore. They're spawning outside of some of these bays and harbors, and the telemetry data, some tagging data done by UMass Dartmouth and DMF kind of supports that. But basically what we found is, you know, Pleasant Bay is basically a giant nursery. You know, it's a really important nursery habitat for a variety of species like these winter flounder that may not be mating in there, but the little ones come in and rely on the eelgrass and the salt marsh and some of these other habitats, those algae beds and mussel beds, as habitat to grow up, spend their first years of life when they're so vulnerable to predators. We also saw things like these herring here. These are not river herring. These are Atlantic herring or sea herring. These are species that also spawn outside of places like Pleasant Bay. But what you'll see here, see how you can see almost through that fish? So that's a newly metamorphosed sea herring. So that's gone from an egg to a larva to what you might call a smolt or something like that. And these little tiny post-larval fish are coming into places like Pleasant Bay and maybe Barnstable Harbor to use it as a nursery. So you can see this is just from one trawl set, but you can see there are some of these immediately sort of post-metamorphosis fish, and you can kind of match them up based on the markings to some of these slightly older by days or weeks fish. So again, you have an offshore species that's coming inshore to use it as a nursery. And again, now this is kind of what I said, you know, there are some species here that, you will use Pleasant Bay or other places for their entire life cycle. So we saw adult squid, we saw those eggs, we saw these little guys. I should have a scale bar there. You can kind of see the wrinkles in my hand there, but that one's about this big. That's a very young juvenile, kind of like a 22 caliber squid. So again, comparing numbers is really, really interesting, from the 1960s to now, but they're really just snapshots in time. It does set a new baseline with which to compare going forward, and that's really, really valuable, and I'll circle back to that theme at the end, but we counted a lot of critters. We found some species that weren't there before. We also found that some species that were seen in the 60s were absent. And that said, I'll talk a little bit more about Nauset in a second, but one of the things we did in Pleasant Bay that was really more focused, so this wasn't kind of replicating methods that were done in the 60s or more recent scientific studies, but really reaching out to the community and trying to focus in on specific areas. So for focusing in on the shellfish, one of the things that we did is, again, kind of going out to the community. And this is a process that we're kind of starting here today, hopefully, but we expect to continue until we're actually on the water. Going out and talking to fishermen, going out with fishermen, kind of getting their feedback. There's Charlie showing me on a chart on my laptop where he was seeing lobsters. Going out and working with a mussel fisherman to see what's there. Taking historical maps of the area and basically just drawing on them with a Sharpie and looking at areas where there's abundance in different places over time. We talked to some of the different shellfish constables around Pleasant Bay and got their ideas about what to look for and where and what they were interested in. And then again, we kind of hit it with a variety of different survey methods. Intertidal quadrats, kind of the very sciencey approach where you randomly throw a square on the ground, dig it up, count what's there. Actual commercial diggers will have to make fun of us when we do this, but it is a nice replicable way of doing things. We also go to the complete opposite extreme and rely on commercial gear. So this is a dredge survey that we did in Pleasant Bay using a small quahog dragger and their quahog dry dredge there, except with a modern twist. We've got this thing covered in cameras, so we could have both a recorded feed and a live feed from the dredge, so we could see what we were missing too. And it gives us some things like this, where we again took a snapshot and looked at things like quahog density around in Pleasant Bay, different parts of the bay, again kind of focusing on areas that shellfishermen told us to look at, or the shellfish constables wanted to know how good their planting was working, things like that. We did something really cool because we're in a shallow system there. We were able to kind of go out and map the edges of these mussel beds. So these black lines, we're basically just going at it relatively low water and either working from a shallow draft boat or on foot using a GPS and kind of drawing lines around where the mussel beds are, because those are kind of ephemeral. They kind of move and change over the years, even over the seasons. I'll show you that in Nauset. And then we would sample those beds and look at size composition and things like that. So that's the kind of more focused shellfish survey work we can do. And we plan on doing some of that in Barnstable Harbor as well, again, with guidance from the community on what we ought to be looking at and where. Switching gears a little bit, I apologize to John and Peter and a couple of other folks who have seen this lecture before. If you were at the Cape Cod Natural History Conference back in March, this is going to be a relatively quick version of that talk, but again, I kind of want to show you some of the tools that we use and some of the things that we can find. So more recently, that was Pleasant Bay, 2014 through 2017. In 2020 and 2021, in this case, the town of Eastham supported a study that was somewhat similar, a benthic habitat map, some fish and shellfish studies, particularly fish that they kind of threw in, but it was really important to look at for them in the context of things like dredging, changes in the barrier beach. And this study was really driven by the geology. Nauset is a really dynamic system like Pleasant Bay, where you have this constantly changing barrier beach system. And again, we had this benthic map that Agnes did. Again, these little numbers here are her benthic stations there. So we picked a subset of those to focus on, as well as revisiting some stations that were done in the 80s. I'll talk about that and skip this slide. It's a little redundant. This is what I just said. We care about these bodies of water. I will just take a moment to say, we care about these bodies of water. And I talked a little bit about some of the different reasons we care, and I touched on why the town cares in this case, but we're living in a time where climate change is happening, ocean change is happening, We have a lot of human impacts on the system, from nutrient pollution, you know, things like dredging, commercial fishing, recreational harvest, you know, all of these different things that are happening in the system. Some have a greater effect than others, but we're affecting the system in a lot of ways, sort of both in an insidious way like climate change, as well as a more physical way like coastal development, nutrient runoff, all of these different things. So we have to take into account all of these different stressors. But in order to do that, in order to really think about what's having an effect on the system, you have to have data. And you know, I'll circle back to this at the end. You know, these snapshots are great, but really this is more about looking forward. Again, same tools, newer boat, same idea. We upgraded for the East Ham study. We've got a Butchie now, it's a nice shallow draft skiff. Very similar methods. We focused on, again, a couple of Agnes's benthic stations and a couple studies that were done, in this case, not by the state, but by scientists at Rutgers University that were working with the National Park Service and the National Seashore, sampling in Town Cove and Nauset Marsh. Same tools, just slightly different areas. We also added for this study a couple of new things that we hadn't been doing before. One was we had video quadrats, so we were dropping camera systems down and looking at the bottom in different areas. We're going to add a twist to this for Barnstable Harbor, hopefully, which is to do these with bait and bring in fish to them and see what we can see on the cameras. Again, in a really nice non-invasive way of sampling. That's another place we'll love to have some volunteers because you got to watch all that video after you collect it. But we'll circle back to that. And we also did some seasonal benthic sampling. So to take Agnes's sort of snapshot and see how it changes over the course of the season. And I think we'll look at doing at least one spot in Barnstable Harbor just to see how her map data changes over the course of the season. Again, same sampling as well. Another new tool that we threw at this, and again, this was in partnership with, started with a high school student that I was working with at Nauset. That's where I went to school, so it's kind of fun working with students and science teachers. And now I'm at the point where it's the kids I went to school with, kids that I'm working with. But it's really, really fun. We started this project as a sort of a summer project with one of the high school students, and this turned into actually a really cool study. And another gear that we started using, these are eel traps. That's my little assistant there. These are just standard eel traps that you can get at a Cape Fisherman's Supply. And they're a really great crab trap. They're all right for catching eels when there's nothing else in there, like a lot of blue crabs and green crabs. We certainly caught a number of American eels with this. But we started as kind of an opportunistic thing. We were trying to get into some of the shoal areas in northern Nauset Marsh that we couldn't get into systematically with some of our other gears. And this evolved into something that we're still doing today, which is monitoring every week in Salt Pond in East Ham. And I'll talk a little bit about some of the things we found there. Getting into the results. So trawling and dredging in these different areas, you know, again, species of commercial and recreational interest like horseshoe crabs and bay scallops, lots of little ones of both. Some cool things that we hadn't seen in this area before, like this little nudibranch, this sea slug, thousands of organisms counted and identified. Green crabs again, lots and lots of green crabs. Here's a place where we have these European oysters, this little spot down by Mill Pond. There's a lot of European oysters in there. So these are a couple of invasive species, although some people say these are delicious. Some people say they taste like a mouthful of pennies. Jury's still out. Hearing more people say they like them than they don't like them now. But again, fish diversity, your Oscar the grouch fish here, some little tautog, other species like that like structure. Again, lots of juvenile blackbacks, little winter flounder, and some tropical fish like these Atlantic moonfish that showed up in Town Cove in the summertime. Blue crabs, that's a species that tends to be resident in Pleasant Bay. They're episodic in Nauset Marsh. They were there in the 60s. They're back again now. They were absolutely everywhere and at all life stages when we did this study. So that's Agnes. She can't be here, but just get a picture of her wrestling a female blue crab. Not sure who's winning there. And again, green crabs. Thousands of green crabs. This is one of my ventless traps, just plugged to the top. Green crabs. So we'll talk about green crabs. You know, the green crabs have become a little bit of a project at the center, and we'll talk about what we're looking at now. But the usual suspects again, the bait fish. It was really cool. We got some of these really tiny little silver signs. These are like barely the size of my fingernail. They only live a year, So with these seine nets, when we do it throughout the season, we'll see multiple cohorts of silverside. We'll see the probably the close to end of life fish that are about like this, and then we'll see the little tiny ones that are just relatively new to the system. They only live a year. They're like the fruit flies of the fish world. I actually study evolution looking at silversides because we grow so many generations of them in the lab, literally like the fruit flies of the fish world. Some other species that were interesting, like this little smallmouth flounder. These are from the seine samples we took. Smallmouth flounder, squid showing up in Salt Pond in Eastham. That was really cool. I didn't know they'd poke their heads in there, but they do. And more tropical fish. So in both Town Cove and I think almost all of these were down in Town Cove here at Hemingway. Oh, not at Hemingway, at Collins Landing. These are jacks, Crevalle jacks. This is the first time I'd ever seen this species on the Cape. Those are mangrove snapper. That's another tropical species that I hadn't expected to see. And all of these tropical fish observations, back when we were working in Pleasant Bay, spawned, bad pun intended, a study of tropical fish. I had a master's student at the New England Aquarium and through UMass Boston. He studied these tropical fish. So we can talk more about the tropicals later, but he's got a whole project based on these tropical fish, collecting citizen science observations of the tropical species. This is really interesting, and this is one of those things, you see these tropical fish and go, oh my God, climate change, and yes, they may be an indicator. Most of these species likely come up when the Gulf Stream, the warm water current that comes up from the southern, has these rings that come off of it that can entrain warm water. And southern tropical species, it kind of spills them off closer to places like Cape Cod, and they'll find their way into our warmer waters in the summertime. They'll stay there. They might try to swim out. They're awfully small, so they may not migrate all the way back down to the southern reef systems where they came. And what they probably do, we don't know this for sure, but what they probably do is try to stick their heads out and see it's colder in there, turn around, and come back in. And so these species likely don't survive past the year. Once it gets pretty cold there. Former student Mike, he actually will collect these and bring them to the New England Aquarium, keep them there rather than going to the tropics and harvesting larger organisms of the same species for display at the aquarium. They can get these little guys that aren't going to make it anyway. And again, kind of getting the most out of the presence of these animals. But his citizen science data that he's collected is really useful because now we're starting to be able to look over the years and see if these things are increasing or decreasing. And this is one of those things. Again, you might look at this and go, oh my God, climate change. There are records of mangrove snapper in Woods Hole from the 1800s. So these things do happen episodically. But that's why I focus a little bit on that historic literature, because it's so easy to see something and think you've seen it for the first time. Not pick up an old text or talk to a fisherman and think this is the first time you've seen it. So we need to be careful getting too hand-wavy about these observations, but they are really, really interesting. And one of the most interesting things we found was this opportunistic sampling that we settled into Salt Pond with. We saw lots and lots of green crabs, and that's actually evolved into a diet study of green crabs. We're now looking at what they eat, and we're looking at the seasonality of when they molt. And we're doing that for a couple different reasons. One is because we're interested in their diet and whether they're eating a lot of shellfish. Green crabs are one of the reasons the soft shell clam fishery has really gone down in Maine. And so we're really interested in what they're eating here, if they're having an effect on cohort recruitment in Pleasant Bay or abundance of shellfish in Nauset. But one of the other cool things that we found using those little traps is lots and lots of black sea bass. And like the blue crabs, this is another species that seems to be on the move. Now these are gradually shifting north. And again, it's not uncommon to see sea bass in places like Cape Cod Bay or Pleasant Bay, but it's really rare to see them throughout much of the year. And we're now seeing these little juveniles showing up in May. As big of a fish as can get into the opening of that eel trap are there from May through October, November, till the water gets too cold. So we're starting to see these sort of juvenile fish setting up shop here. It'd be really interesting to see if we're seeing these in Barnstable Harbor. Maybe you are already. I look forward to hearing from some of you. I know there's folks that are potting sea bass out in Cape Cod Bay. They're certainly here, but I'm really interested to note that Barnstable Harbor is kind of a nursery for these little ones. To summarize, we take all of this information, we publish it in scientific literature, we publish it in scientific reports. you know, we'll circle back to that. We compare to some of the historic studies that were done in the system. We can also take the data that Agnes studied. We can kind of stack ours on top in different ways. So this is a kind of an example where we have 3 different methods of looking at one place. So this is one station, number 16, out by the inlet. This is an area where there are these sort of very dynamic and ephemeral mussel beds. So that here they are seen from underwater video. Here they are seen dumping the dredge on deck. And here they are seen at a very micro level under Agnes's microscope, looking at mussel seed. And when you look at the camera data, you can see how the density and the size composition of mussels varies throughout the year. And when you compare all of these methods, you start to get a real sense of the ecology of these different spaces. So that one station, just looking at blue mussels, you know, you can see the different size composition. So lots of small ones here, lots of big ones and macroalgae, some dead shell, and lots of sea stars, which love to eat blue mussels. So you're getting an account and a sense of the predators here too. So you've got predators and prey, you've got animals growing and changing in their size composition and relative abundance during the season. So these are the kinds of things we can get at by pooling the dredging and trawling, as well as the cameras and Agnes's benthic work. So really building on the work that she's done for you all and that she's done in other places. We'll fast forward past this again. You know, we report stuff out. You know, we'll create a technical report for the friends, like we did for the benthic study. We also publish this information out of the scientific literature. That's really important for scientists to publish this stuff, but also for everyone who's supporting this work to have that recognition that there's peer-reviewed research out there that says what's living in here. Little baby horseshoe crab there. That said, so we talked about 2 different snapshots, Pleasant Bay, Dawson Harbor. You know, those are really great, and they provide a really, really valuable baseline. And I think the work that we're preparing to do in Barnstable Harbor is going to do the same. But those snapshots, when you compare them 50 years apart, like we did in Pleasant Bay, you know, they're only so valuable without using that information to design a framework for long-term monitoring. So to look at the relationships between species, we're going to go back to some of these places that had such interesting ecology, interesting species diversity. You know, we already started long-term monitoring in Salt Pond because that's an easy lift with students and volunteers and AmeriCorps and so forth. But, you know, I'll use this quote from Terry Pratchett, you know, in the context of looking at these snapshots and things like that. You don't know where you come from, then you don't know where you are. You don't know where you are, then you don't know where you're going. You don't know where you're going, you're probably going wrong. And I love that quote because it talks a lot about, you know, the value of looking at historical information, the value, more importantly, of looking ahead and thinking about, you know, where we are now, where we've been as much as possible. But if you don't have data points in between 1965 and 2015, it's really hard to make inference about the causes of change and variability. So one of the things that we hope to do with the data that we're collecting with you all is we'll find some of these areas, both based on your input, based on the data that Agnes has collected and that we'll collect, and look forward. You know, we can say, okay, this area and maybe some of these species are really important to monitor, and this method worked really well to measure and monitor their presence in abundance. And you know, going forward, we'll be able to actually understand the causes of changes in places like Barnstable Harbor that are so important to us. I think with that, this is a great time to kind of pause, take a moment to grab a coffee or a snack, and then we can come back and kind of talk about going forward, what we can do in Barnstable. Got to open the floor for questions and discussion about where to go next. Does that sound good?
Sounds good. Cookies are in the back.
Oh, yeah.